So in this lecture segment, we're going to learn about two processes. One is called protein secretion, in which the organelles are going to work together to accomplish something. And so the first one is shown here, and then the second one is called intracellular digestion. So let's go over protein secretion. Inside the cell, what you have here, just want to highlight what we've got. They're showing you the nucleus here, the nuclear envelope, and they are showing you some ribosomes. Those little red dots are on the outside surface of the outer nuclear membrane. And then continuous with that in light blue, they're showing you the endoplasmic reticulum. Keep in mind, they're showing you a section so you can sort of see inside the space inside the endoplasmic reticulum. In real life, it's not sectioned, it's complete, but the way they've sectioned it in the image is so that you can see that there's a space on the inside. So almost as if somebody has sliced through it. And so what you can see is that on the outer surface, we're still inside the cell, but on the outer surface of the endoplasmic reticulum are those ribosomes. So this light blue is the outer surface of the rough endoplasmic reticulum, and then the darker blue is indicating the inside space of the endoplasmic reticulum. And the inside space of any compartment is sometimes called the lumen or the cistern. I won't use the term cistern, but sometimes you see that. But lumen, L-U-M-E-N, is the inner space of one of these cellular compartments, and sometimes I just say space. So the endoplasmic reticulum has lumen, and what happens is that the ribosomes that are on the outer surface are making proteins, and those proteins are threaded through pores, holes, in the endoplasmic reticulum, and the proteins end up inside of the endoplasmic reticulum space. So I'm just putting some little proteins in here. Oops, that one is not in the right place. And so let me erase that one. So then what happens next, the proteins are made by the ribosomes, and they're threaded through these pores, these channels, and they go into the endoplasmic reticulum lumen. And at different places on that membrane, there will be little sections that sort of push outward on that membrane, and the membrane surrounds a section of the contents of this endoplasmic reticulum lumen. The membrane kind of creates a little pouch, and then it continues until it touches right here. It will eventually touch its back to itself, that membrane piece. And that little piece of membrane will separate from the main part of the endoplasmic reticulum, and then it's just a little sphere holding some proteins inside. And so when it's separated from the endoplasmic reticulum as this little sphere of membrane containing, in our example, proteins, we call it a vesicle. So a vesicle is what separates from the endoplasmic reticulum, and it carries the proteins from the endoplasmic reticulum, and it travels until it reaches the Golgi. So a vesicle is going to carry macromolecules from the endoplasmic reticulum to the Golgi. So this is ER, and this is Golgi down here. So the Golgi are shown in orange or yellow, whatever color you think this is. And when the vesicle gets to the Golgi, that membrane actually fuses to one of the Golgi compartments, and the membrane becomes part of the Golgi, And whatever was inside of that vesicle gets dumped into the lumen or space inside of the Golgi. So the vesicle is a transport vehicle to get materials from the ER to the Golgi. And in the Golgi, you'll remember that it acts as sort of a post office, sorting the molecules, macromolecules out, the ones that were delivered from the ER, and then putting them in separate piles. And then new vesicles will bud off of the ends of the Golgi and carry those macromolecules to different destinations. Sometimes they go to the next Golgi compartment, or they go to a further Golgi compartment. Sometimes they go off to other destinations, but still inside of the cell. And in our example, we're going to talk about vesicles that ultimately come off of the Golgi. So look at this one here. It's going to come off of the Golgi. Let's say it's got a certain type of protein now in it. And that vesicle will then be carried to the plasma membrane, and it will fuse to the plasma membrane, and that membrane becomes part of the plasma membrane now, and it dumps whatever was inside that vesicle is dumped out. So it releases the contents of that vesicle. So vesicles are carrying proteins. In our story, vesicles are carrying proteins that were made at the ER by ribosomes. And then from the ER, they enter into vesicles, which then pinch off of the ER and travel to the Golgi, fusing to the Golgi and dumping that mixture of proteins into the Golgi space. Inside the Golgi, things are happening. The molecules are being sorted. And sometimes and most of the time, they're being modified. Modified means when you modify a molecule, it means you're attaching something to it, or you're cutting something off. So whenever you see that a molecule is modified, it could be a protein, could be a carbohydrate, could be DNA, RNA. And we use this word modified, it means attaching things to it, like could be functional groups. It can also be trimming things off. So modified. So the Golgi acts to sort the molecules, modify the molecules, and then send them out to their final destination. In our example here, when we use the word secretion, it means that the molecules are being released from the cell entirely. So there are proteins that are made, all proteins are made inside of cells because they're made by ribosomes, which are always inside of cells. But some proteins need to leave those cells and go out into the body to find the place where they can do their job. A good example would be insulin. Insulin is a hormone, it's a protein. It's made inside of pancreas cells. It's made on the ribosomes on the ER of the pancreas cells, and then it's moved by vesicle to the Golgi, where it's modified, and then it's moved to the cell. It's moved out of the cell by vesicle, released from the cell. So we say secreted. The protein is secreted. And it needs to travel in the blood to find its target cell. But it's just an example of a protein that has to be made inside of a certain type of cell, but then released in order to find its purpose, its function in the body. So that whole story is protein secretion, the making of the protein, moving it through the ER, through vesicles to the Golgi, and then through vesicles to be released from the cell. So it's a process. It's something cells do. There is an organelle that actually pinches off of the Golgi, just like a vesicle, and it's full of digestive enzymes. So digestive enzymes are hydrolytic, meaning they cause hydrolysis reactions. So the word digestion and the word hydrolysis mean the same thing. So there are hydrolytic enzymes. We haven't quite gotten to what enzymes are, but those are proteins that make reactions happen. So there are hydrolytic enzymes that are made in the ER sent to the Golgi, sorted, and then they pinch off in vesicles, but the vesicles don't really go anywhere right away. They're just waiting. So a lysosome is a vesicle that was pinched off of the Golgi. It's full of these hydrolytic enzymes. I'm just going to write HE. And it's waiting to do what it needs to do. A lysosome has a function, and so when those lysosomes form, they're full of these hydrolytic enzymes. So then the question is, what is it that they do? Well, they destroy cells and they break down food matter, food molecules that the cell has engulfed in a process called phagocytosis. So it gets us to our second story, our second process, things that cells do. I don't particularly like the diagram that's provided by your book because I don't think it's as easy to follow. So I'm going to make a bad drawing over here. So the lysosomes in my story have already pinched off of the Golgi. I'm going to put them over here. Here's a lysosome. It's a membrane compartment. It's an organelle surrounded by membrane, and it's full of hydrolytic enzymes. I'm going to put HE. Those are proteins that break down by hydrolysis macromolecules. And so what the lysosome is waiting for is molecules to break down. And the way it knows what is supposed to be broken down is that the cell will bring in the molecules in a process called phagocytosis. So outside the cell, I'm drawing the cell membrane here in red, there are molecules. And what they do is they come to the surface of the cell from the outside surface, and the cell surrounds them with membrane, and it pinches off inward as a vesicle. So this is food that is brought in from the outside of the cell. So anytime there's a food vacuole, the lysosome, which I show here, will fuse with that food vacuole. So these two will combine together. And what it makes is a bigger compartment in which the hydrolytic enzymes are now mixing with the food, and they're breaking it down because hydrolysis breaks polymers down into monomers. And so if we wait after enough time, there won't be really any food in here anymore. There will just be monomers. There won't be any macromolecules. There will just be little monomers. So little amino acids that were part of some proteins that were once in this vacuole, little monosaccharides, little fatty acids, different monomers will be inside this compartment now. And what the cell does is it releases them across the membrane into the liquid portion of the cell, which remember was called the cytosol. So those monomers are just leaked across this membrane compartment into the cytosol. And so that's how the cell digests things. It brings in food particles by this forming of a vacuole around it. This process down here is called phagocytosis. We'll get to that in a later chapter. But the bringing in of the food particles, phago, CYTOSIS. Sorry, that's so messy. Phagocytosis, this is a G. And that's the bringing in of the food into a vesicle, or we say vacuole. And then that food vesicle or vacuole fuses with the lysosome, which contains those hydrolytic enzymes. And the mixture of the food with the hydrolytic enzymes causes the polymers to be broken down into monomers. The monomers are then allowed to pass, oh, I didn't spell cytosol right, into the cytosol. Y'all probably noticed that. That's what I get for talking and writing and thinking at the same time. So that's how the different monomer units get into the cytosol. Then what's not part of intracellular digestion, but the reason the cell does this is now it has monomers available and it can build new macromolecules from the monomer subunits that it brought in and digested down. So this is how the cell gets its monomer units. It brings them in and then breaks them down from polymers to monomers and then releases them into the cytosol to use. So this is called intracellular digestion. If there's anything left in this compartment, anything that was large that could not be broken down, what happens is the cell simply moves that vesicle back to the membrane. It fuses with the membrane and those undigestible components, molecules, are released. That's waste. So intracellular digestion is a process by which cells bring in macromolecules and break them down into monomer units. The other story is protein secretion, where the cell is making a protein at the endoplasmic reticulum and then moving it by vesicle to the Golgi and then moving it by vesicle out of the cell. So two different things that cells do commonly: protein secretion and intracellular digestion. So I'll have some practice questions where you're going to put the events in order. First this, then this, and then this. So you'll get a chance to practice putting the story in order, I guess you could say. All right, a couple other organelles that the cell has, or that most cells have. One is called the peroxisome. Peroxisomes are important for breaking down what we call fatty acids. You may recall fatty acids from our chapter on macromolecules. A fatty acid has a very long chain of just hydrocarbon and then a carboxyl group at one end. Oxidation means the complete combustion, the complete destruction of fatty acids. And the reason a cell does oxidation is to get energy from. So the oxidation of fatty acids is the breakdown of fatty acids as part of the process to get energy from those molecules. But what happens in the peroxisome is that hydrogen peroxide is made. We call it a byproduct because it's sort of one of the unfortunate products of the oxidation of fatty acids. And hydrogen peroxide is poisonous to cells. Fortunately, it's not allowed to exist very long because the peroxisome has a second enzyme called catalase, which detoxifies or makes the hydrogen peroxide harmless. And what it really does is it converts hydrogen peroxide into water. So there's inside the peroxisome, there are reactions to break down fatty acids, which is good. That's how, if you're trying to lose weight, fatty acids are part of those triglycerides. And so the triglycerides can be broken down and then the fatty acids can be broken down. That's how your body breaks down fat. But when your body is breaking down fat, you produce this hydrogen peroxide, which would be poisonous, except you have in your peroxisome catalase, which detoxifies it. So it's a whole system. It works really well. And then the next structure is called the proteasome. It's a big complex. It's a big structure made of protein. And the funny thing is, it's made of protein and its job is to break down protein. So that's kind of interesting. It's made of protein and it breaks down protein. So any misfolded, we talked about unfolded proteins, anything that's damaged or when things get old need to be recycled are broken down by the proteasome. So part of the recycling of old molecules, if you will, or structures that are made of protein. And the cell breaks them down into amino acids and then reuses the amino acids to build new proteins. Plant cells have a couple additional organelles. They have a very large compartment surrounded by membrane. It's called the central vacuole, and it's full of water. And when you water a plant, what you're really doing is causing each cell in the plant to fill with water in its central vacuole. And that helps the plant to it puts pressure on the cell wall of each plant cell, and that's how the plant is able to stand upright. If you don't water a plant for a while, one of the things that happens is the central vacuoles don't have very much water, so the cells are kind of shrunken a little bit, and the plant will become floppy, or we say flaccid. So the central vacuole in plant cells holds water. Now, this is probably your favorite organelle of all time, the mitochondrion. Mitochondria is plural. Mitochondrion is singular. But in any case, all cells that are eukaryotic have mitochondria. There's a bit of a confusion, I think some people think that plant cells don't have mitochondria, but they do. All eukaryotic cells, everything in the domain eukarya, has mitochondria, usually lots of them. And it's important for the metabolism of the cell, cellular respiration. So I want you to highlight the word cellular respiration. I know that you learned that mitochondria are the powerhouse of the cell, but we're going to kind of let go of that definition and put a bit of a more college level definition in, and mitochondria are where cellular respiration happens. I'm not going to give you any more definition right now because chapter 7 is all about cellular respiration. So for right now, we've just got a few things here that we want to learn, that mitochondria are found in all eukaryotic cells, and they're responsible for this thing called cellular respiration. What's interesting about mitochondria is they have their own chromosome. In addition to, of course, the nucleus in the cell has chromosomes, but each individual mitochondrion in that cell has its own chromosome. And they also have their own ribosomes. So inside each mitochondrion, there are ribosomes, separate from the ones that we were looking at earlier when we were talking about protein secretion. There's ribosomes inside of the mitochondria. So that is interesting, and I'm just going to leave it there for right now, but it will come up later as having a significance. All right, chloroplasts. Chloroplasts are important for photosynthesis. So just like I said, we're not going to learn too much about the mitochondrion right now. We're not going to learn too much about chloroplasts right now because photosynthesis is the topic of Chapter 8 lecture. But it's an organelle in plant cells, and there are some other eukaryotic cells that have chloroplasts. But plants are going to be the main group that we're going to focus on. And there is a pigment inside of chloroplasts called chlorophyll. So chloroplast is the name of the organelle, and chlorophyll is a pigment molecule found inside of the chloroplast. And so chlorophyll is a molecule. And it's a pigment, which means it has you can detect a color when you look at it. And it's important for this process called photosynthesis that we'll talk about in Chapter 8. So just connecting a few things. Simple question on your follow-up quiz, your post quiz. We'll just touch on these things. They, like the mitochondria, they have their own DNA, they have their own chromosome, and they have their own ribosomes. So there's something special about the mitochondria and chloroplasts. Not only do they have these special functions for respiration and photosynthesis, but also there's this interesting thing where they have their own chromosome and their own ribosomes. Okay, so I'm going to take a break because this lecture has gotten a little long, and I'm going to come back with the next segment, which we will start with this endosymbiosis.